physiology. Microbial physiology is defined as the study of how microbial cell structures, growth, and metabolism functions in living organisms. It covers the study of viruses, the bacteria, fungi, and parasites. It is also conveyed as the study of how microbial cell functions, which includes the study of microbial growth, the microbial metabolism, microbial cell structures. The microbial physiology is important in the field of metabolic engineering and also the functionals of genomics. Now, we will move on to metabolic enzymes. So what is metabolic enzymes? We all know that enzymes are familiar with us. So here we will discuss the metabolic enzymes. Metabolic enzymes are loosely defined class of enzymes that regulate metabolic pathways in energy homeostasis, including glucose, lipid, amino acids, metabolisms. Metabolic enzymes often serve as drug targets for metabolic diseases such as obesity, insulin resistance, diabetes, and cardiovascular disease. So, the role of enzymes in metabolism. Some enzymes help to break down large nutrients molecules such as proteins, fats, and carbohydrates into the smaller molecules. Each enzyme is able to promote only one type of chemical reactions. The compounds on which the enzymes act are called subrates. So, in enzymes, there are three types of enzymes. First is the amylase. When we say amylase, it is the other carbohydrates enzymes break down the starch into sugars. Second is the protease. Protease is the enzymes that break down proteins into amino acids. The last is the lipase enzymes. When we say lipase enzymes, it is the breakdowns lipids such as fats and oils into fatty acids and glycerol. Metabolic enzymes carry out a variety of cellular functions necessary for survival and homeostasis, including proteolysis and digestion, the cellular respiration, energy storage, transcriptions, and response to the environment. These life-sustaining pathways are vital for growth and maintenance of cellular integrity. Metabolic enzymes encompass a wide range of different protein classes, including carboxylases, dehydroxylases, lipoxygenases, oxidoreductases, kinases, and lyases, and transferases, and etc. So now we're going to metabolism. Metabolism is a term that is used to describe all chemical reactions involved in maintaining the living state of the cells and the organisms. Metabolisms can be conveniently divided into two categories. So these two categories are catabolism and anabolism. So guys, when we say catabolism, it is the breakdowns of molecules to obtain energy. So in anabolism lawat, it is the synthesis of all compounds needed by the cell. So in anabolism, it kinakailangan ating mga cells. So metabolism is closely linked to nutrients and the availability of nutrients. Bioenergenetics is a term which describes the biochemical or the metabolic pathways by which the cell ultimately obtains energy. Energy production is one of the vital components of metabolism. It is the process by which your body converts what you eat and drink into energy. During this complex biochemical process, calories in food and beverages are combined with oxygen to release the energy your body needs to function. So, metabolism kinakailangan aton energy, aton body to function. So the three main purposes of metabolisms are: first, the conversions of food to energy to run the cellular processes. Second, the conversions of food fuel to building blocks for proteins, lipids, nucleic acid, and some carbohydrates. Last is to eliminate of nitrogenous waste. So that the three that I said earlier is the three main purposes of metabolism. So these enzymes catalyze reactions, allow organisms to grow and reproduce, maintain their structures, and respond to their environment. So guys, the word metabolism can also refer to the sum of all chemical reactions that occur in living organisms, including digestion, and the transport of substances into and between different cells, in which case the above described set of reactions within the cells is called intermediary metabolism or intermediate metabolism. So, in bacterial genetics. Bacterial genetics is the subfield of genetics devoted to the study of bacteria. Bacterial genetics are subtly different from eukaryotic genetics. However, bacteria still serve as a good model. For example, animals' genetic studies. So the study of how genetic information is transformed either from a particular bacterium to its offspring or between interbreeding lines of bacteria, and how that genetic information is expressed. There are three methods of genetic transfers in bacteria. First is the three mechanisms of horizontal gene transfer in bacteria. First is the transformation, second is transductions, last is conjugation. The most common mechanism for horizontal gene transmission among bacteria, especially from a donor bacterial species to different recipient species in conjugation. One of the major distinctions between bacterial and eukaryotic genetic streams from the bacterial lack of membrane, the bound organelles. Like other organisms, bacteria also breed through and maintain their characteristic from generation to generation. Yet at the same time, they exhibit variations in particular properties in small proportions of their progeny. Though heritability and variations in bacteria has been noticed from the early days of bacteriology, it was not realized then that bacteria to obey the laws of genetics. Genetic information in bacteria and many viruses is encoded in DNA, but some viruses use RNA. Replications of the genome is essential for inheritance of genetically determined traits. Gene expression usually involves transcriptions of DNA into messenger RNA and translations of RNA into protein. One of the major distinctions between bacterial and eukaryotic genetic streams from the bacterial lack of membrane, the bound organelles. Like other organisms, bacteria also breathe through and maintain their characteristic from generation to generation. Yet at the same time, they exhibit variations in particular properties in small proportions of their progeny. Though heritability and variations in bacteria has been noticed from the early days of bacteriology, it was not realized then that bacteria to obey the laws of genetics. Genetic information in bacteria and many viruses is encoded in DNA, but some viruses use RNA. Replications of the genome is essential for inheritance of genetically determined traits. Gene expression usually involves transcriptions of DNA into messenger RNA and translations of RNA into protein.